Okay, welcome. This is my first YouTube video ever. I was asked by a friend to record some of my theories about COVID long haulers and SARS-CoV-2 virus and put them out on YouTube. And so I am going to give it a go. First of all, I'll mention that I do have a Recover from Long COVID webpage. And I also have a Recover from Long COVID Facebook group that I've started. I guess it's at the end of December now, and we're at about 200 people. And the purpose of this is mainly just for me to share my personal protocol of how I have gotten well with fellow long haulers in the long haul community in general. I'm not claiming that I have a magic bullet. But I can say that my second long haul, I was quite sick at the end of October. Had a hard time. I was so fatigued I had a hard time getting into the shower. I could walk about 100 feet and that was it. And I started experimenting with quercetin and resveratrol and various other ways to induce something called cellular autophagy. And to my pleasant surprise, it seems to work, at least for me. And I am now, basically 8 weeks later, I am 95% recovered. My only lingering symptom is a little bit of insomnia, and that is improving. So anyway, let me go over my thinking on this. And I do have a scientific background. I have a Bachelor of Science in Cell Biology, and I was trained as a PhD in cellular immunology and with a lot of broad scientific research experience. Although I've been out of the scientific research field for a couple decades, so I'm kind of diving back into the science and the COVID-19 pandemic. And learning lots of things, that didn't exist 10, 20 years ago. Okay, my working theory is that long COVID involves persistence of the SARS-CoV-2 virus infection and at some point persistence of viral debris. And electron micrographs, when they go into like the intestinal gut epithelial cells and look for the virus, one research group has found viral particles present in the gut even after 6 months. And when they look at the micrographs, they see the little virion particles, and they also see what are called protein inclusion bodies, which are basically just aggregates of misfolded proteins, likely aggregates of misfolded viral proteins. And these aggregates can be very tight and linger around for a long period of time. And I think that's part of the problem is to get rid of those outside of our cells that have been infected by the virus. Okay, like every virus, the SARS-CoV-2 has a bag of tricks to co-opt normal cellular machinery and maximize production of viral particles. So basically it invades our cells and uses our cells as a copy machine. And I believe one of the tricks is to upregulate cell metabolism by activating mTOR. And mTOR is just a protein complex in our cells that's like the central regulator of cell metabolism. It's like, imagine it as a lever, and if you push it forward or you activate it, it upregulates cell metabolism, turns the cells into the high growth mode, and the virus likes that because it gets to crank out the maximum number of viral particles. Whereas if you take quercetin, that seems to like pull the lever back and put the cells more in a neutral mode. And then if you add resveratrol, it binds to the mTOR complex, and it it actually inhibits the mTOR activation function. So what you're really doing is you're triggering a whole ancient set of cellular genes or pathways that evolved probably, in response to a variety of stress responses. When you fast and you're facing starvation at the cellular level, this autophagy is triggered, and it causes degradation of proteins within the cells, and it can even degrade damaged organelles such as mitochondria, which produce the energy for your cells. And so it's kind of like cleaning house or repairing your house. It's like, okay, we got to hunker down, we got to get nutrients and resources wherever we can, and it's an ancient cellular process that was identified, or it was recognized for its central importance in 2016 by the Nobel Prize in Physiology and Medicine. Went to the Japanese scientist that first elucidated the whole process of autophagy in yeast. So this mTOR, when it's forward, it's like your maximum growth mode, and the mitochondria are working really hard, and part of a result of that is you get reactive oxygen species bouncing around, basically oxygen molecules or oxygen free radicals bouncing around the cell, bumping into proteins and lipids, and just reacting with whatever they bump into and damaging them. It's almost like rusting the cells from the inside out. And also the virus co-opts the cellular translational machinery, so like the machinery to crank out proteins, and up to 60% of the mRNA messages in a cell, in a SARS-CoV-2 infected cell, can be devoted to encoding proteins for the virus. So that's a huge percentage. It just totally takes over. Off all autophagy. So basically you pull the lever back, like in fasting or other cellular stresses, autophagy is triggered, and other genetic programs are repair and stress resistance programs are triggered, such as the sirtuin genes. For example, sirtuin-1 is instrumental in DNA repair. Some of the other sirtuins are involved in mitochondrial repair. And when it's neutral, there's a little bit of basal autophagy going on, perhaps. But when you move that mTOR regulator of metabolism, full speed ahead, metal to the pedal, stepping on the gas, basically autophagy is totally shut down. And I believe this is one of the key ways that the SARS-CoV-2 virus hides from our immune system, because autophagy, the cellular degradation and recycling process, is key to degrading viral proteins, chopping them up in little bits in the acidic lysosome, and then little fragments of the protein get presented on the cell surface on these specialized MHC molecules for surveillance by the immune system cells. So specifically the cytotoxic CD8 plus T cells are constantly scanning the surface of our cells, trying to distinguish between self and non-self. And if they see a fragment of a viral protein, then they'll take appropriate action and either kill that cell or sound the alarm and cause inflammation and recruit other immune cells to the area. So my theory is that because the virus, if the virus is able to strongly activate mTOR, it shuts down autophagy. That helps it hide from our immune system. And by triggering autophagy, either by fasting, for example, fasting from Friday evening to Monday morning, that basically triggers autophagy in Saturday evening and Sunday evening. and or via these other mechanisms. I'll talk about that in a separate video. But basically, every time we trigger autophagy, there's this degradation of proteins and organelles, damaged organelles within the cell, and this antigen presentation of the viral fragments on the cell surface. And what I have discovered in my SARS-CoV-2 infected long haul body is that on a very repeatable pattern, whenever I induce autophagy, in the evening, typically in the evening, sometimes at night, I experience a transient inflammation. The first time I felt it, I was doing an extended water fast, and I felt just a pressure in my heart. I mean, I was alarmed enough. I was thinking about going to the hospital or the ER to have it. I wasn't sure if I was having a heart attack or not. And I had that pressure under my sternum for about 4 or 5 hours in the evening. And then it gradually subsided until they went away. The next evening, I had the exact same thing happen, except for this happened for like 3 hours, and it was not quite as severe. It was never real painful, but the pressure was less. And the third evening, I just felt it for about an hour or an hour and a half, and the pressure was milder, or in other words, the inflammation was less. And then actually on the fourth evening, I could barely feel anything, and it didn't last very long at all. So after that, I was curious to see if I was fully recovered, and it turned out I was. Anyway, the idea is that every time we trigger autophagy, a percentage of the viral particles are degraded. It's just our lysosome, they get surrounded in a lipid membrane, and that fuses with the lysosome, which is the garbage can of the cell. It's acidic, and it breaks down the proteins into fragments and eventually into individual amino acids. So, and each time it pinches off just a percentage of the cellular cytoplasm. You can't remodel your entire house in one weekend. You could maybe make progress remodeling your bathroom or your kitchen, but remodeling everything takes time, and it's kind of the same with your cell. To degrade and rebuild and remodel the cell, it does it a little bit at a time. So I'm estimating, maybe 15 or 20% of the viral proteins get degraded each weekend cycle of autophagy, and that's kind of what I advocate, is triggering the autophagy just on Saturday and Sunday each week, and then allowing your cells to recover and rest and rebuild, Monday through Friday, because that seems to be a good cycle. You really do need some recovery time from triggering autophagy. Yeah, so what's really cool about this is this antigen presentation and trait, of the viral proteins. That tells us exactly where the virus is hiding in our bodies, because this SARS-CoV-2 virus, it can infect the endothelial cells of our blood vessels, and basically this virus has access to almost our entire body, many different organs, very many different tissues, and it's kind of random in what tissues and organs it infects in a particular long hauler. So for example, my second long haul, it hit me harder than the first time, and my first autophagy experiment, I was taking quercetin, 800 milligrams of quercetin, and I had read that resveratrol can induce cellular autophagy in cell culture, so I took 300 milligrams of resveratrol, and that's a compound kind of a plant stress compound from grape seeds and grape skins and some other plants. And to my surprise, that evening, well, actually that early that night, I could feel my kidney. I could feel inflammation in my kidney. I was laying on my back in my bed. It woke me up. It wasn't terribly painful, but I could feel pressure, my lower back, kind of close to my spine. And what was really weird was that I could feel it pulsing. I could feel it pulsing with every beat of my heart. It would be like the pressure would be a little bit greater, and I could just feel this pulsing in my right kidney. And I had no clue that the virus might be in my kidney. But because of this antigen presentation and this transient resulting inflammation, the inflammation and the pressure told me a virus was in my right kidney. And I'm like, wow. And then after 10 minutes or so, I turned over and went back to sleep. And well, actually, at the same time, I also had an increased gut ache. So I believe the virus was in the epithelial cells lining my small intestine. And I also had an increased headache, so I believe the virus was actually in my brain. That's certainly what it felt like, and that's consistent with, I think, with the brain fog and the headaches and the migraine headaches that a lot of long haulers get. Fortunately, autophagy happens in every single cell, or as far as I'm aware, every single cell, every single tissue, every single organ in the body. So no matter where the virus is at, even if it's infecting neurons in our brain, by triggering cellular autophagy, we can little bit by little bit degrade and get rid of the virus. I'm now convinced that the Achilles heel of the SARS-CoV-2 virus is that it's susceptible to cellular degradation via triggering autophagy. So in my group, Recover from Long COVID, I have this autophagy protocol, and I outline different ways to trigger autophagy and have some simple guidelines for fasting and when to trigger autophagy, how long to fast. And basically, I recommend that individuals limit their autophagy induction to just 2 days a week, Saturday and Sunday. And then I have I've put together through Microsoft Forms and kind of the cloud, a simple symptom survey that takes 5 to 7 minutes to fill out. The first time you do it, just say initial survey on question 7, and it'll take you through a few more background questions. And then every Thursday and Friday, I'm asking people to take this symptom survey and just catalog what symptoms you had that week, and if you got better or worse, or if you had any new symptoms or concerning side effects. But it's my fervent hope that over time, it might take a month or it might take several months, that I'll be able to collect enough data to start presenting, to start having evidence whether or not this autophagy protocol actually works for people other than myself. And I'm hoping it does, and I'm hoping to get evidence showing, full recovery or significant improvements in a lot of long haulers, so we can get engage some medical researchers and clinicians and get some, double-blinded, placebo-controlled clinical trials going on this. But regardless, I just feel like it's kind of up to the long haul community to experiment and try different things, similar to the early 80s when HIV hit the U.S. and the world. It took a number of years before effective treatments came out, and it's nobody knows exactly what to do right now. And through my personal experience of 2 long hauls, I think I've kind of accidentally stumbled on something that works. And so I encourage people to join my Recover from Long COVID Facebook group and take the weekly surveys and track your symptoms. And if you recover on a timeline similar to me, I'm anticipating that most long haulers will be fully recovered in about 12 weeks, 10 to 12 weeks. Thank you very much for your time, and I'll do some more videos. Good day.